Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker for pharmaceutical advertisers, and now I'm the president and a member of Biotech Without Borders, a nonprofit based in NYC dedicated to aim at the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've mostly worked on bacteria, but I've also worked in research integrity, and I'm currently working for a scientific journal.
Every week, we meet to talk about microbiology, and today it's our coolest microbiology papers that we've seen during the week.
Yeah, and at the end of the month, we'll be selecting a paper. So at the end of this month, we're going to be looking at a paper on bioink, right? That's going to be exciting.
Yes, microbial ink.
Yeah, brilliant, to print nanofibers, so very exciting. But obviously, if you have any suggestions for papers you want to cover in extreme detail, then tweet us at microtwjc if anything has caught your fancy.
Absolutely. You can follow along with the papers that we discuss on this week or any others in the Shards of Terror library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. And boy, do we have a show for you today. This has been a long time coming. Hopefully our sound is good, because we've had a couple of production delays because of the past 2 weeks. Our equipment and trying to improve our things. But now we've got like a cavalcade of papers, things that we've culled from the past month. We're looking at some new papers on the clinical severity of BA.2 and also reinfections and original antigenic sin and also like an animal model of long COVID, which very exciting and interesting stuff. Plus we've got other things outside the realm of SARS-CoV-2. We've got magnetic bacteria. How do they work? We've got a virus that could make you smarter or dumber depending on how you look at the data. We've also got like there's a structure of algae. There's a new type 3 secretion system new secretion system. Sorry, I got it wrong. There are so many types I get mixed up. And of course, when we talk about all things small, something that is much less small than usual, a centimeter long bacteria, which is completely unusual.
Yeah, I think it's been a challenge to only have 10 papers like in the time that it takes to cover in an hour. But yeah, hopefully people are going to be interested to see what we've collected. And of course, at the beginning, we're still in the middle of a pandemic. Of course, in many countries, the rates have gone down, but it's still out there, and we don't know what's coming up next. What we've experienced is Omicron, and the first paper that we're going to be covering, or preprint rather, is clinical severity of Omicron sub-lineage BA.2 compared to BA.1 in South Africa.
Yeah, that's right. So the past month, there's been a lot of talk about BA.2. So Omicron, it created this massive spike, and that's been decreasing. But people have been noticing that BA.2 has been taking over as one of the sub-lineages of Omicron. So the question is, like, is this going to be a new peak? Is this going to this lineage of viruses going to be more dangerous? And that's what this paper aims to look at. So this paper is a study based in South Africa, and it looks at the national COVID-19 case data and a bunch of like laboratory data and data from DATCOV, DATCOV, which is an active surveillance system for COVID-19 in hospital admissions. And so what they did is they took all this data that they've been collecting throughout this outbreak, and then they've isolated the strains that they think are Omicron and the strains they think, Omicron BA.1 and BA.2. And there is one key difference between these strains. So BA.1 has this thing called S gene target failure. So it's got this deletion that means certain PCR tests mean that some PCR tests turn out differently. So they don't have like an S gene in them because of the way that...
I think we've seen this before, or at least like this has been the focus of other sort of typing typing things, because Delta also showed up in the way where the S gene wasn't able to be replicated.
So Alpha, I think, was the
Alpha. Okay.
I believe that was the UK strain. And then Delta could be told because that didn't have the S gene target for knockout. So we've almost like had this way of differentiating strains because they have this mutation on off, and BA.2 doesn't have this mutation. So that means through between them, because one of them, sequence a bunch of genes and one of them turns out without that S gene, that's probably Omicron BA.1. And in this situation, that means that the ones that have all the S genes in is BA.2. I'm not explaining it especially well, but yeah.
Well, this is they call it a proxy, right? Right. This is not the way that they're differentiating between BA.1 and BA.2 in this particular study is just on the identity of this S gene, right? Whether or not it's S gene positive or S gene negative. And the reason why that supposedly works is because, right, the current circulating strain when they were doing this was S gene negative, right?
Right. Yeah, that's absolutely right. And I've got pulled up a graph that kind of shows the transition between them, because transitioning from a time when Omicron was dominant in South Africa, and that was, and then BA.2 slowly taken over. So you can see the changes in S gene positives over that time. And so
it's gone down.
Yeah. So what they decided to do is look at the cases in this time frame and whether they're becoming more or less severe.
By looking at hospitalization.
Yeah, that's right. So they looked at severe disease was defined as a hospitalized patient meeting at least one of the following criteria: admitted to intensive care, receiving any level of oxygen treatment, ventilated, or other treatments, and of course if they died. So that's obviously the severe symptom of COVID. And what they found was that the odds of being admitted to hospital did not differ between either BA.1 or BA.2. And so what they kind of are coming to the conclusion is that while BA.2 might be able to outcompete BA.1, it doesn't seem to be producing a more severe disease, which is, I think, pretty good news. And
I feel like that's everything.
Yeah.
It's a pretty short thing.
Yeah, just starting off with a short paper, but starting off on a positive note. I feel like good news is something that we need right now. Yeah, and also,
sorry.
Go ahead. I was going to roll on to the new paper.
Go for it.
Okay, all right. So the new paper: Occurrence and Significance of Omicron BA.1 infection followed by BA.2 reinfection. So this is another kind of concern that people have had, was whether there's going to be another spike in cases because of BA.2, and we haven't seen that so far. What we've seen is that there hasn't been additional spikes, and this kind of paper is looking into whether if you've been infected with Omicron BA.1, you're more likely to be infected by BA.2 compared to just the original strain again. So this looks to see whether BA.2 can actually overcome immunity to BA.1.
Yeah. So I just want to add in, I think the big spike of Omicron was seen in South Africa first, so a lot of eyes were on that particular wave of infection to understand what's going on, and they did during their big spike in infection. It started off as BA.1 and then it became BA.2, but the kinetics of that spike still went up and went down, even though the proportion of genomes, right, that they were coming up as BA.2 was just going up the whole time. So that I think observation is what drives people to say it doesn't seem like it would extend the wave or anything, but of course that's just one sort of piece of evidence to get at that. Knowing reinfection could give you this, right, would give us an understanding of whether or not, if you've been infected already at the beginning of the Omicron wave, right, maybe if bound, you just get it reinfected by that, and that would extend sort of the wave, right? It wouldn't crash down. It would maybe have some tailing end. And so, this is the type of question that they're trying to get at by looking at reinfection data. And this is from Denmark. They have seems like an amazing surveillance system in the sense that they're doing genome sequences here. So this is not just a proxy with the S gene target failure, as we were talking about in the previous paper, but instead they can actually build a phylogeny of the viruses that they're collecting.
Yeah, we've actually, over the past month, we've really seen a lot of papers from Denmark that have told us quite a lot about BA.1 and BA.2. Which has been really useful. So yeah, they've got a really big national microbiology database and national patient registry and national vaccine, and they kind of pull in quite a lot of data here. and so they get about I think 1.8 million SARS-CoV-2 cases, and then based on that they screen that reinfections, which is what they just define as people who had positive tests between 20 to 60 days apart during December 2020 and 2021. So that's the big like time frame from the Omicron wave, and from that they could select paired samples. So that's samples from the same person, and then they also selected controls, so people who were just randomly selected to compare, and then from that they picked out the whole genome sequencing, and in total they found like 263 cases with paired genomics data. And from that they could screen out to see like who was firstly infected with Omicron and BA.2. So they found like 47 cases that had that reinfection. And for comparison, sorry.
I want to point out in this one, like, it's kind of crazy to me that 443 samples failed sequencing. So like they had a potential 1,000 data points, right, to draw from this, but methods fail, right? I guess this is like frozen samples probably inside of some sort of biobank.
Yeah,
I guess.
I feel like that. I mean, because this does happen does happen regularly when you're sequencing genomes, I feel, but when you're sequencing that many, you don't necessarily. These are mostly for like, these are biobank samples, so I don't know what the deal is with that, but yeah, that does seem like.
Yeah, it just means that like even with like this amazing infrastructure, like they potentially had all this set up. Like I just like for me, I like to point out, right, that there's so much of science where it's like, yeah, you can like have all these big numbers and these big plans, but it comes down to like how you get things done at the bench sometimes. And you can't account for some of those things, and that will leak and make the data set a bit weaker. But again, like this is still an amazing set they're able to bring together. The fact that they have the infrastructure to bank these things and then do whole genome sequencing, like, yeah, I'm excited to see what we're learning from it.
And one of the things that I found quite interesting that I'd like to draw your attention to is that they looked at in their cohort they did find like they compared like say Delta to Omicron reinfection and found like there's quite a lot of cases of that, and they also looked at the ability of the people being infected by the original strain, so Delta to Delta, BA.1 to BA.1, and they found like 19% of cases. So that kind of being reinfected with the same strain, I'm seeing as the control here. So
yeah, I think they wrote in the discussion a little bit that at least in the BA.1 to BA.1, when they have the sequences from both, and so like they can see sometimes the sequences aren't too different. So that could be not a reinfection, but rather just the residual, based on like the definition in which they pulled things out.
I think you got a good point there. I think I might have been jumping the gun on that part of the article. But yeah, they did find that reinfections are occurring. So and they do some characterization on that to look at, say, symptoms to see whether. I think they tend to find that the symptoms are less severe the second time around.
Yeah, so it can happen.
Yeah, it can happen.
It can happen. But they also point out that like when they look at that cohort, right? So that's 47 of the 263 cases, right? That they show BA.1 is the initial infection, BA.2 is the second one. Most of those, a large proportion of those people are unvaccinated.
Yeah, that's right.
Yeah. So I mean, again, people always say like, or like this was a thought that occurred like early in the pandemic. It's true, like natural infection gives you a different antibody profile, right? I think people think it gives you like a breadth profile instead of a depth profile in terms of neutralizing antibodies. But at least in this instance, right, when we're looking at BA.2 infection, like, yeah, BA.1 wasn't able to give the unvaccinated a good complement of antibodies, right, in order to fend off the second one. I guess probably it's kind of just right. This is the challenge, I think, too, of data in retrospective, right? It's like we can't control these groups to like ask the questions that we want. Like these are observations. They're all observational data, and we're limited by like the bias of sampling here.
Yeah, that's true. So yeah, reinfection does occur. I'm not necessarily convinced it occurs, say, more more frequently than more
Frequently, yeah.
than like
Then,
the original infection. But then again, you did raise a good point about how some of those original infections could be long term. So it,
yeah,
but I
yeah.
think the important point is it is a high degree lower than say the reinfection from Delta to Omicron, which has been the driving factor in this outbreak.
Yeah. Yeah, like the difference, right? I mean, we just know this, right? I mean, I think people know this by now in the news. Like Omicron has all these immune evasive abilities, and those apply to BA.1 and BA.2. So that's why it's become so successful. Yeah, whether or not BA.2 will drive something. I mean, to me, again, I think we've seen the observation that BA.2 isn't
Yeah,
driving a second spike, but that's also in the background of all the different interventions, right? Like non-pharmaceutical interventions that were rolled out during the time of the Omicron wave, which are now being rolled back. So
yeah,
yeah, I feel like that's, like that to me, there's a big uncertainty there to know where the effect is coming from. Because again, retrospective study, we're just sort of like looking at observations from behind, but there's many confounding variables that go into, right, the conclusion that gets drawn out from data like this. So yeah, I guess I'm hopeful that like this means that BA.2 is just, whatever, it comes to dominance. Some people get reinfected, but that's not a normal thing. But we don't really know in the sense of like predicting the future. Restrictions get lifted and then we're going back to normal. Maybe we'll see more. We'll have more. I mean, this sounds horrible, but like if people are throwing caution to the wind and having more exposure events, then we'll begin to gather that data set of whether or not reinfection is going on with BA.2.
So well, I guess we're going to have to hold our breath a little bit because we don't then.
Yeah, I think so.
I mean, well, I'm hoping that there isn't going to be a spike once we lift all the sanctions, because that basically means that okay, it's over now, we can celebrate. But I am always concerned.
At least until the next variant rolls around, right? Like I think that's like in terms of the pattern that we've seen throughout the pandemic, right? There's been like a spike, people get a break, and then if there's not vaccine equity, then all of a sudden a spike comes again from somewhere else.
Yeah. So I think that's our BA.2 news, but we have still got some Omicron news.
Yeah.
So this is a paper I wanted to talk about for basically a month, and it's so this is about the Madonna vaccine. So they've created a version of the Madonna vaccine that is tailored to Omicron, and so this is a study in macaques to see whether this Omicron can be used as a booster, and whether that could produce antibodies that are protective or neutralizing against Omicron. And
Yes. Yeah, we originally trotted this paper out 2 weeks ago in like our robot glitchy voice episode.
yeah,
And I feel like I hadn't done a good skim of it at that point, but I skimmed it a couple times since then. And it's interesting. What I picked out was that they had done this with the beta as well in like previous studies, that they had sort of looked at a full course of vaccination plus booster. And the reason why they went down this path of the Omicron is because they thought it was very hopeful in the beta case that a beta specific booster was able to boost neutralizing antibody titers or whatever other metrics they were using for efficacy.
I mean, on that same week we did see a mouse study which seemed to also be very positive on boosting but this is in rhesus macaques, so they're kind of closest to humans.
Yeah.
And what's interesting is that they find that so it's not so much like they do find that there is an immune response to Omicron, but I think the thing that is interesting is that the kind of immune response they get is that it's reactive. So so they characterize like the B cell specificity after boosting, and following like the the Moderna vaccine, I think that they found like lots of B cells were specific for the wild type alone, but when they boosted with Omicron, they found that there were cells that were like they reacted to Omicron, but none of them were specific solely to Omicron. So so essentially this is something that is called antigenic imprinting, or original antigenic sin, if you want to be very alarmist about it. But the idea is that when your immune response is first devised, it reacts to the initial insult. So when you're first teaching the immune system to work against SARS, that first strain is going to be its template for all the other interactions it's going to have, which means that when it gets hit with a version of the virus that's very different, it's not going to go back to the drawing board and create a whole new immune response. It's going to tailor that current immune response to that virus. So what you're going to find, what they found here is that the Omicron reactions tend to be cross-reactive rather than really specific to it.
Yeah, so boosters work, right? Is the takeaway, right? Like it does increase to get a booster, but when you compare boosting with the regular version or boosting with the Omicron version, they're kind of the same in terms of the efficacy that they bring. I guess the initial vaccine, right? Like sort of what you're saying with this antigen sin thing is that like the original vaccine run is what's giving you your initial like way of responding, and it's hard in the Omicron case, at least it's hard to push it towards a more specific Omicron thing.
So I think this is and the thing is it works both ways. So that means that if you say vaccinate someone with just Omicron, then the concern is that that version will respond to Omicron very well, but if say any strain comes up that's related to the original strain, it's going to be less good at doing that. And so now we have this kind of concern. Do we want to create a vaccine tailored to a strain that is less severe than the original strains, at the risk of making people a little more vulnerable to the original strains, or do we keep vaccinating people with the original strains? And in the hope that Omicron doesn't evolve further. So this is a challenge.
Yeah, 2 weeks later, right? Like we originally had this on the docket for 2 weeks ago, and at that point I was like, why did they even bother doing this? I hadn't read it that deeply, right? Like now I know, like they did it because they saw promising results previously with another boosting regime. But maybe like now I'm like almost thinking, because the Omicron wave is so low, at least where I'm living, like my mind has like sort of shifted in thinking about this paper as like maybe this is actually more about like the abstract this concept that you're talking about, right? Of like what is the best way to approach, right, making yearly vaccines, or like things where we know that there's change happening. Maybe there's information in this paper that that speaks to you know the flu vaccine or whatever, just in an abstract fashion, right? In like design considerations. But yeah, we won't be seeing. I think the takeaway though is we won't be seeing an Omicron specific booster.
I think that
both because like there's no it's not working as well. Tool production, right? They already have tons of boosters. And at the end of the day, I think I still want to say this, and I think I said it 2 weeks ago, is that boosters are great, like tool, like they're kind of like a Band-Aid tool, right? Like it's like, oh my God, like things like rates are going up, and we want people to have that extra layer of protection. But you know, that Swiss cheese model of protection, it's only one layer of Swiss cheese. There are many other ways to prevent like a big breakout from happening. So yeah, what's more important is definitely getting vaccines to people who don't have them at all. Right, that's going to make the biggest dent.
Yeah, I agree. And so now we're going away from the Omicron papers, and we're going to be looking at
SARS-CoV. No, it's still
Omicron paper,
SARS-CoV-2,
which is Omicron triggers cross-reactive neutralization FC effector functions in previously vaccinated but not unvaccinated individuals. So this is an interesting one because one of the things we've been looking at when we're studying Omicron is neutralization of antibodies. So that is if they stick to an antibody, antibodies will stick to the surface of a virus, and then that should prevent the virus from functioning. And so when we're looking at things that will hit the core points of the spike protein. So things that will hit the receptor binding domain or the But the thing is that antibodies have more functions than just like blocking the activity of whatever enzyme they bind to. They also act as signaling molecules for the rest of the immune system. So the thesis of this paper is, well, we don't know exactly. We know that antibodies against Omicron are less, say, good at neutralizing. But what about all the other things that antibodies do? And so,
uh-huh. Which is like wild, right? Like I think this highlights the way in which science can be very narrow in that when it asks questions, and like we hope that our viewers listening to us like are sort of building up their implicit understanding of that, in that you only get the answer to the questions that you ask specifically. So like neutralization assays are very specific for this phenomena of antibodies like basically gumming up the works and not letting a viral fusion event to occur. But that doesn't address this whole other function of antibodies, which is, right, they have a Y, and the back part of the Y is the FC part, which immune cells recognize, so they can say, oh my God, like here's something we should eat. Or like shoot like weird cytotoxic molecules at, right? Like there's sort of many different signaling pathways that FC effector can work down. But yeah, and they look at the assays that they're using here, I think, capture some of those. It's phagocytosis is the one that they look at, and then also cellular toxicity. So yeah, one of those is like, do they eat the thing once they touch the FC, or do they shoot out like cytotoxic molecules when they touch that FC receptor?
Yeah, and I think what they find, there is a difference between people who've been vaccinated and unvaccinated in terms of, say, just the spike binding and I think antibody dependent cellular phagocytosis.
Yeah, and yeah, it's kind of so. Yeah, I guess. Yeah, that is what they find.
Yeah,
I don't really have any more to say about that actually. Like,
and
like the spike binding, it's kind of interesting to see that the spike binding has like a non-significant. I mean, there is a difference there in spike binding with Omicron, vaccinated versus unvaccinated, but the difference is really pronounced with the cellular phagocytosis and the cellular cytotoxicity assay.
Yeah, and I think another thing that is really interesting is how cross-reactive these responses are. So I think the Omicron triggered antibodies are very cross-reactive for all variants tested. So that kind of it kind of it follows on from the previous study.
That's a good news thing though, right?
Yeah.
That's a good news idea.
So
like just in general, yeah, Omicron reactive antibodies are helping build up protection in other ways to other variants out there.
Yeah. So yeah, and like I mean, yeah, it's yeah, that's basically the good news aspect of it. That
yeah, but like this is on the background of like we've always, when we say like better, it's like in this abstract, it's a very abstract like way of talking about it, because we're looking at like these cellular assays and we don't know how that translates into disease protection and like transmission protection necessarily, right? It's good. We know that it's on the side of the host. These are host protective mechanisms, we think, mostly. I guess like even that is a little bit, that can be kind of, that can actually be kind of blurry as well, whether or not these are necessarily host protective.
And like when people vaccinate, and also I think the thing they looked at was people who were unvaccinated and their first infection was just Omicron, and their responses were less cross-reactive to other strains. So that again, like goes back to what we were previously talking about, antibody imprinting, and how in this case it seems like having the original version of SARS-CoV-2 as the imprint seems to be better for gives broader protection than, say, if you just gave Omicron as the imprint.
Yeah,
but of course this.
Yeah, I mean, I was gonna say like I hope we don't. Maybe I do. It'd be great if we have listeners that are thinking like I got my infection with Omicron and that's giving me protection. Like, don't think that. that's not a good line of thinking. Like, get your vaccine.
I mean, but like to counter that, I think that like these are with past variants. So we don't know the future variant. We don't know where it's going to come from.
It's true.
It could bud out from Omicron, in which case we know that we've got some protection against it. But if it comes back from like a version of the wild type, then people who are infected with Omicron who are unvaccinated before they got Omicron might be in a state of more vulnerability. So
Right.
that might be something to
Well,
watch out.
this is like a really complicated. These are the types of questions that we'll be these are the very complex public health questions that we might be grappling with, right? Should a new variant appear, right? Like, what would be the policy changes in different ways in terms of defining vaccination, defining protection?
Yeah, so I
and so
feel
forth.
like it's kind of like a herd immunity situation where people who have been vaccinated and maybe had an infection, that they should be more prepared, but then it affects a small percentage of the population as
Mhm.
a pandemic goes on. So I don't expect that. I feel like there was less pressure on governments to put to put up protections for people as the
Mhm.
number of people who are unprotected gets lower and lower. So that's
Mhm.
my kind of concern, because
Yep.
I because sometimes I'll be talking to people who are in that kind of diagram box of people who are vulnerable, who can't get access to this kind of protection. So in the ideal situation, be able to
Yeah.
control outbreaks, but it's
Well, I think we must say now at this point, right, that like autoimmune, right? People who are immune compromised, people who are dealing with immune issues, like they're very vulnerable during a time, right, where there's no control of like community spread. So yeah, I just want to make sure people know that all this chatter is on the background of that as well.
yeah. So I think I've said all things I set out to say.
Yeah.
But well, Minref has a question. Uh, but what if they're trying to fill out the SARS-CoV-2 reinfection loyalty card?
Oh dear.
Oh no, I'm sorry, I got this new setup, so or because you before only like Dan could read the comments, but now I can see them as well. So this is a whole new world for me. Yeah.
Yeah, smack my head. All I can say.
Yeah, you got to catch them all. Just like Pokemon.
Well, good reasons not to catch them all is in the next two papers. Still SARS-CoV-2. This one is actually, I think this one is a little bit to try to rectify. I wanted to bring this up because like we had covered, right, a lot of the preprints and things looking at severity of the Omicron variant. So this paper is Hospitalizations of Children and Adolescents with Laboratory Confirmed COVID-19. This time period, right, span, but includes before as well. And it's quite a simple study in the sense of it's just like looking at who goes into the hospital in this particular age range. But I think understanding that this age range, children and adolescents, they're less vaccinated because like it took time for the vaccinations to be approved, right? And then of course, like once approved, then it takes time to like get people actually interested in being vaccinated. So understandable, the lag sort of exists. And what they see is in that Omicron predominant period, as the cases go up, they also see like an increased spike in the hospitalizations amongst these age groups. So both the 0 to 4, like extremely in the 0 to 4, the primarily unvaccinated group, because there aren't vaccines approved for that age range, but they also see that bump in 5 to 11 and 12 to 17, which are included, but in the vaccination range, but less. So the reason I brought this out was just to give people a sense that we did not, at least in the States, and maybe this holds out in other countries, did not do a good job at flattening the curve, as we talked about like at the very beginning of the COVID pandemic, right? That was like a big concern. Not doing a good job of flattening the curve means like what happened was many people ended up in the hospital because of it. And it did hit this age range that maybe people weren't thinking about as being vulnerable necessarily to COVID. So yeah, I just wanted to point that out. This is like kind of in a hindsight. This is a very hindsight type.
Yeah,
paper.
I think that this is good because generally, like I think we did come out with there are a couple of papers describing about why children are less susceptible to COVID-19, but I think people almost took that as less susceptible and immune as the same thing, but it isn't. And
Yeah.
so when the Omicron outbreak came out and so many more people got infected. I don't think people were expecting as many like hospitalizations or deaths as, and this could have been prevented if we had more vaccinations in young people. But I think like
yeah, more vaccinations or fewer community infections,
yeah,
right? Like they're sort of like, right, they're both strategies to try to mitigate these things.
yeah, and I think there's been a lot of like talk around vaccines and their safety that has prevented good rollouts happening in younger children because of that are preconceptions that they are less vulnerable. So it's really unfortunate.
I just want to make sure, and again, like I think we can move on right after this. Like the main thrust, I think, of presenting this data to everybody is just so that we can digest. Like Omicron, like the wave just passed in some ways. It's still, in some places, it's still going on, first of all, right? Like to check your local numbers, right? To know like what's actually happening. But like it's passed in many localities, but it's good to digest what happened because it just happened in many ways. And just like because like if we do see another spike, it's good to go into that with like the knowledge that we have from all the previous things that have happened, and we want to like have sort of an accurate view of what that means.
For sure, yeah.
Okay, that's all for me. So punching through to the next one, we've got an animal model. SARS-CoV-2 infection results in lasting and systemic perturbations post-recovery. And this is great. This is these lasting and systemic perturbations, aka right, long COVID or something like it, right, is the hope that this model is going to shed some information on. And we've seen this. We actually saw this before. I don't remember when. Definitely in a news episode, they were using the Syrian hamster model as a way of looking, like we know that they have a closer ACE2 to us. So like the SARS-CoV-2 establishes an infection, a lung infection in them that's more similar to humans, I guess, than in mice. And then in a previous paper, I saw that it depends where you look at in an animal model, right? You can learn different things from it. So I think the last sort of like, I would say like expansion of this model that we saw was the cardiovascular effects, right? Like let's focus in on the heart and let's see like, because we know like SARS-CoV-2 does a bunch of things to the heart. Let's see like, can we see some of those mechanisms by focusing on the heart? But here they're like, well, let's look at some other places that might be like long lasting. They look at the brain. And what I really enjoyed about this one is they use like a comparator virus, a comparator respiratory infection on this one, right? Where they bring in the flu. And so like I really like the way that they brought together right these tools that we have at our disposal, like experimentally. We have an animal model, right, that has these similarities. We're able to compare what a flu infection would look like versus a SARS-CoV-2 infection and get at that sense of like both create acute respiratory illnesses, right? That means the immune system is going to be responding in a certain way, but only one of them, well, what they discover in this paper is only one of them kind of then goes on to create like gene expression changes in the brain. And then they link that. Oh, sorry.
I didn't want to interrupt you. You had a really good flow there. I mean, for me, this almost feels like a direct follow-up to one of the first papers we did on hamsters, where they found out that hamsters like their nerves were getting, and that was as like kind of an explanation for why there was a loss of sense of smell. I think that's been brought.
Yes.
So I think there's been a lot of similarities in the way that SARS-CoV-2 attacks hamsters to humans. So they are emerging as like a really useful model. And this paper does go deep. There's a lot of like histochemistry in this, where they take slices of the hamster brain and take you through like what's happening to the tissue and like the kind of inflammations that you're seeing.
And again, just to reiterate, like they have controls from influenza infected hamsters. So like this is not just like, oh, these hamsters got sick, right? Like this just happens in the brain. No, this is like this virus is doing something in this context.
I think that's an important point, because like there are defined sickness behaviors and things that happen as a result of just general inflammation that make you more groggy. And less able to do. Actually finding stuff that's
Right.
distinct
Right.
and as like kind of for SARS-CoV-2 only, that's a really important function.
Yeah. And they do tie it up at the end with a little bit of a behavior test as well for these hamsters. They look at this buried food, time to find buried food behavior. And right, that's supposed to be this surrogate for, like, I guess, brain fog and like cognitive issues, right, in humans. And they do see like this 3 days post-infection buried food. It takes those animals that were infected with SARS-CoV-2 longer.
Yeah, and some hamsters
this.
never find the buried food. That's just so sad. They don't know how that.
Yeah.
But yeah, so it kind of puts in. I mean, obviously these are animals. They're not gonna. They shouldn't go one-to-one with humans, but they think they do give us a guidance as to how SARS-CoV-2 behaves in a different organism and how that might affect that organism. And from there, we can kind of try and pull out what we can apply to the human situation.
Yes. Yeah. So I think importantly, I actually don't really think about what I see the contribution of this paper is it's giving us the ways to measure, like the things that we can measure in the model. But now we can dive in and we can say, how does SARS-CoV-2 do these things, right? And like understanding the how might give us like maybe like an immune correlate that we can then go back into humans and say like, is that happening in humans? Does that mean that we can do something, right? Like to help people, like maybe this model can help people find therapeutics for those who are experiencing long-term symptoms from COVID. And that would be really exciting. And we know that hasn't been highly reported. The reporting on that has been like in the background this whole time, right? But like we're definitely going to see more and more of that increase. And it's important that we have the tools to help people in that sense.
Yeah, I think this brings, for me, this brings long COVID almost into my like sphere of understanding, because quite a lot of that research is very much in the psychology and neuroscience areas, which are, as a microbiologist, like kind of very complicated and outside of my sphere of expertise. Whereas when you start bringing into like immune correlates or like kind of stuff you can see under a microscope, and you can see there's damage there, that makes it a lot more understandable for me. But when you're looking at, say, much broader things, where you're looking at questionnaires and neurological tests, that's quite complicated, and it's a lot harder for me personally to figure out what symptoms mean. And so this paper has kind of given me an into that kind of world.
Yeah, absolutely. No, I think that's like it's important. Like this is a great model. Interested to see it being published. This is only in preprint right now. Once it gets published, I think it'll be nice to see like the commentary around it and certainly follow up studies. I think like that just help us understand the mechanisms behind some of these long-term effects that you might have that people are reporting that they experience after COVID infection.
Yeah. Right. Okay. Oh, that's the end of our SARS-CoV-2 coverage, and now we're moving on to some really amazing papers that we found over the past month.
Yes, it's been so hard to whittle these ones down.
Yeah, there's been so much good research, but this is the one that we're going to be choosing. Let me see if I can post this.
Yeah, so this is from 2 weeks ago. It was on our list for a while. We wanted to bring it up again just because like we haven't talked about magnetosome organelles before, and we want our audience to put it on the radar, right? We'll probably see other papers about this in the future, but they are inclusions of mineral inside of membrane bags. People call them invaginations of the bacterial cell membrane, and that wasn't totally known. I think it was only sort of discovered recently. People thought, oh, it was actually just they thought the minerals were just like sitting inside of the bacteria and floating around the cytoplasm, covered in membranes, of course.
I think there's this big thing about bacteria where like people didn't believe the bacteria had organelles. I think that's another thing that like in like university in like school, I was basically taught, bacteria, they just have this kind of matrix. They don't have like any like all together. Yeah. But we're finding out
it's just soup. Cytoplasmic soup.
I think we're finding out more and more that there's a lot of order in there, and that some bacteria do have tiny organelles like eukaryotes, and magnetosomes are one example of that.
Yeah, they're membrane bound. They have a membrane around them, but I think they're also open to the environment because I think that's probably where they get the source to grow the crystal, right? Like they have to have some like external minerals to get in there. So anyways, these are organelles that are found in bacteria. They end up aligning to a magnetic field, and it's thought that the function of them is to allow these particular bacteria to move to favorable conditions. So they essentially, it's their private compass that they use to orient themselves to the world, and so that they can go to their favorite chemical gradient.
Yeah.
And the question is how do they grow? Like, what are the genetic factors? You know, people want to understand this basic information. Oh, this picture that you're getting.
Yeah, this is
Oh yeah,
a
this.
3D model they based on like various kind of slices through the bacteria, and you can see these
Mhm.
magnetosomes, they align in a line basically. So that
Yeah.
so and
which which is a great assay if you think about, like, so from the genetics mindset, you're like, oh wow, there's a clear phenotype here, right? Like magnetosomes lining up. And so you can go in and you can start like breaking genes and seeing like what ends up breaking this successful phenotype. And using that information, dive deeper into some of those regulatory mechanisms. And that is what they do in this
yeah.
paper.
So
I think they know the candidates though. They have like a cluster of genes that they're focusing on breaking.
yeah. So they've got some genes that are focused on like so you've got this membrane that's growing, and inside you've got the actual magnetic kind of
Crystal, yeah.
crystal. And in this picture, the crystal's in purple and the membranes are in yellow, and the rest of the cell is
Hmm.
in blue. And I think they break things to see, like, firstly, how big the crystals can get, because I think the
Yep.
question of like, do the crystals get big and push the membrane out larger, or does the membrane become larger and then that helps it grow the crystals?
Hmm. Hmm. Yeah, so they make a bunch of different mutants and they see it has different effects, and they build up that information into a picture. I think they end up zeroing in on just like a specific event, this MAMD protein that is membrane bound. And they find out if they disrupt it and it can't get into the membrane anymore, that sets the system off, right? Things don't develop properly.
Yeah.
And yeah, I think that's what I want to say about it. Mostly just to show people like these magnetosomes and let people know like these are in bacterial cells, right? Like bacteria are like really quite amazing, and maybe interestingly, right? Like this ability to go in and then like break things in bacteria in order to figure out what those genetic controls are. It gives us this way of it gives us this way of extracting out the regulatory and genetic mechanisms for biological phenomena that is the basis in many ways of biological engineering, because like you have to understand that whole thing to then build it. And yeah, we have sort of amazing tools at our disposal within bacteria in order to gather that information. Yeah, I love, right.
Yeah.
Anyways, I'm
I
just like
mean, I was
gushing about like how I love
I,
bacteria as a
I just
model.
wanted to gush about like how magnetosomes themselves are just such a clever system for bacteria, because a bacteria these are for
Uh-huh.
like certain bacteria that want to have just the right level of oxygen. So there's points in the ocean where oxygen is depleted and is low, so they want to be at a
Mm,
certain depth. And in order to do that, you can either like try and figure out about how much pressure there is, or but these bacteria figured out the most efficient system is just to like have a set of magnets with them and follow a field line, because of the geomagnetic field of the world, it kind of like follows perpendicular to the Earth's surface. So they just have to follow a field
mm,
line in order to maintain the same depth, which is just a very
mm.
clever little system. Um,
Yeah. Oh, that yeah, that is cool. I have this like general sense of it. I'm glad that you dove in a little bit deeper to figure out how the
well,
the magnet is actually
I
giving a proper
think there's
readout.
some sort of is some like kind of different. I think there's still some mystery there as to like how it's how
Hmm.
like say the sensing systems work, how it like how that translates into movement. But that's kind
Yep.
of like, because I remember that was explained to me in like a set in back in undergraduate, and it was like so fascinating. Um,
Yeah. All right, so that's
Yeah.
magnetosomes. Scientists are still figuring out some of those genetic underpinnings. Hopefully, we learn a lot more, and maybe we get to discover them in other systems. I don't know, create our own. There's a lot of options.
Yeah, and now the next paper. Caudovirales bacteriophages are associated with function in memory in flies, mice, and humans. So,
So that title is
yeah,
intense, I would say, right? Like, definitely, it's always big claims when people associate things, right, in humans, right? Like, we know that we don't have the same experimental tool set inside of humans as we do in other model systems, and so it can be incredibly difficult to really dive in and prove, I'll say, right, without a doubt that these phenomena are happening. But that's the advantage of models, right? You can go into models and you can try to validate the stuff that's going on. In this paper, though, I mean, it does seem very interesting for sure, but at least in the way that they describe this paper, like it seems like what they do is they do like associations, right? They're kind of finding correlations within a human set, and then they take those correlations and they bring them into the models, right, as a way of strengthening like their case that this correlation might be real in some ways. And a problem that I have with that is a little bit of like, there's a type of confirmation bias, I think, that's being accentuated when you go down an experimental design like that. So I would definitely say take some of these take any conclusions with some grain of salt. You do have to think about the relevance of these models to the human system.
yeah. I think there's always like a challenge with with correlation studies where there is always a risk of getting like say spurious correlations or like because I guess like this is like this study has a lot of like they take participants and give them a lot of tests about their higher brain functions, and then they catalog their their phagome, and then they try and like correlate which kind of microbiome correlates to whichever function tests they use. So
Mm-hmm.
so this is uh and and so, like, I think that they're in their like first like cohort, they go out, they find some associations between, say, Caudovirales and one or two of their higher functioning tests, the mental tests in participants. And I mean, and think the danger of these studies is obviously like even if you find a correlation, it doesn't necessarily mean that the bacteria or virus itself is the reason for it. Um, the for one example of say like if you say find someone who's eating say who has a lot of agar degrading bacteria, they're eating a lot of
Mm-hmm.
seafood or like they're eating a lot of sushi, which is very
Yeah.
expensive. That could indicate that oh they're more smart, or it could indicate oh they're actually rich and they regularly eat lots of expensive food. Then and that's why they have so the reason why they test high on your high function test is because they've had all this other things happening outside of the microbiome that are affecting them and allowing them to have a microbiome that has agar degrading bacteria because they've got a more elite diet. Um,
Mm-hmm.
so it's that kind of
Yeah, like those are in broad statistical terms, right? Those are the covariates, right? That you can't really account for sometimes. So yeah, it's just difficult to tell. But I think that the experimental design that they sort of try to the way they try to get around that is then they say, oh, but we revalidated that phenomena in an animal model. But like, but the animal model, sorry, you're gonna,
no.
you're gonna,
I was about because they do like a discovery cohort where they do this and they find like positivity on a certain test, and then they do a secondary cohort where they find like that. So they find that people test positive on a different higher functioning test, which is kind of like they've documented their own failure to replicate in this paper, which is kind
yeah.
of bold, but I appreciate it. Um,
Yeah. Um, so So I think that's all we want to say about this, right? Like, I mean, they do some interesting work. We think they do interesting work in the Drosophila, right? Because they they have like they have like this, like now they're not like associating things anymore, right? Like they can actually like do like these, you know, behavioral tests, and like they can change up the microbiota. They can actually do an intervention in a way, right? That shows if it has any impact on their phenotype. But yeah, it seems still like a stretch, I would say, like on how that then how that relates to some of these other.
Yeah, I kind of feel like if it was only the Drosophila study, I'd be fine with it. And also where like, extraordinary claims require extraordinary results. So the idea that a virus can provide someone with a gain of function is very extraordinary. Like if it was the other way around and they said we have a virus that's associated with people being less smart, that'd be more believable to me, because breaking things is a lot easier than building things. But I feel like the way that this paper has been written out has been almost designed to give me the worst reaction to it, even though there is some worth in it in terms of the Drosophila stuff.
Yeah.
And like, so this is why I'm trying my best not to soak this in a cloud of negativity.
Totally. But I think with that we can probably
move on.
Yeah, so this one is fascinating to me. You said, yeah, it's type 6 secretion is what they're finding here. Right. This is a structure of thylakoid-anchored contractile injection system in multicellular cyanobacteria. So yeah, the injection system is this type 6 secretion system. It exists inside of many bacteria. People, I think, well, on the pathogenic side, it exists, right, to inject effectors into cells, but it also exists, people think, to inject toxins into other bacteria as like a bacterial competition. That's where we generally think of type 6 secretion living. But apparently, I didn't know this, but like they can also find them floating in the environment called environmental contractile injection systems. So there can just be like these syringes in a way, like proteinaceous syringes floating around in the environment. And yeah, so like and here they zoom into an algae, cyanobacteria, and they find, oh, these systems exist inside of their the thylakoids, which are the structures that are doing this electron transport chain. Yeah, photosynthesis, electron transport chain photosynthesis. They find these type 6 contractile systems in them. They look at them, they find them using electron microscopy. They have a very distinctive shape.
It just shows you some neat structure within them. And yeah, these videos are always stuck in the supplementary material. It's so sad that they aren't like upfront, but
yeah. Because they're really cool.
It gives you a lovely like fly through of like the structure. Really fun to have something like that.
Yeah. And again, those are all like reconstructions from taking slices. So they have like they have imaging, right, that allows them to take slices. It shows places that are electron dense. And if you stack them up all together, you can create these models.
Yeah, it's very fast, cool. And I think we're gonna like that's taking us to the next thing, which is a
Oh wait, I'll say my last words on this is just, but people aren't really sure what they do, right? Like at the end of this paper, it's like, but what does this do? A little bit uncertain, right? They do show that they become exposed when the cells die, something like that.
Right.
So maybe, right, there's something that happens in the environment that the cells get released and then they, I don't know, inject evil things. Maybe it could be this, right? This is my hypothesis, just randomly thinking like, oh, if something is killing off the cells, what if that is another cell, right? And then they release these little bags with injection systems attached to them. Those like act as like to whatever, neutralize the threat. But it could be, who knows? It could be all sorts of, that's like one random thing. They have no evidence to So in that way, that's what it does.
But yeah, it does look like something that like, oh, an amoeba's eating us. Let's explode and send out like some things to attack it.
Right. But it could be like injection systems are also used for swapping genes, right? Like sometimes, sometimes like you can inject into different type 4 secretion, agrobacterium, right? Right? In that way, they're injecting genes and trying to transform something, I mean, for their own benefit. So like, I don't even know, right? Like there's many ways in which you could have advantages there. Yeah.
So the last paper, and one that I'm very interested in, is a centimeter-long bacterium with DNA compartmentalized in membrane-bound organelles. So this is an interesting one. So so it's been known that there are some like self-reducing bacteria that can become very big. The thiol-reducing bacteria. So they can become. Let me put this pull up my notes because like the largest bacteria before this paper was found to be like 750 micrometers, which is very big in bacteria terms. Most bacteria are in the term about like 2 micrometers, which is a millionth of a millimeter. And the largest one they found before this paper was like nearly a millimeter. So that was like enough to kind of see with your naked eye if you really concentrate. And so that was like, so when I read this paper, the idea of a centimeter long bacterium, that's insane. That's
wild. No, I think there was I remember like people used to talk about some sort of like, there was a large one that people thought of. They only thought it was one. It was like a big like penicillin. It was like, it's some sort of like large fisher bacteria. Do you recall this? In like the guts of fish. I had heard one story before this for sure, but definitely not so much detail.
Yeah. This they found that there's like so in this case they were looking at in mangrove swamps for bacteria. So like to quantify like what's going on in them. So they let me see if I can pull up the picture. But they found like these bundles of like bacterial colonies that could be and so they sampled them and found that these kind of tangled webs of, so kind of looking like hyphae looking things. So there they are, right?
Yeah. Oh, they're strange. And this is light microscopy.
Yeah.
Right. So yeah.
You don't usually see things very well with light microscopy in bacteria, but here they did see some structures. And yeah, what they found was that these are, so they did some like, so they took some out under the microscope and they preserved them using, I think, various fixants to look at them under the electron microscopes. They've got some 3D images I'll show in a, actually I'll pull them up in a bit, but yeah, this is, they did some like various bits of genome analysis on them, and found that, yeah, they are bacteria. And they have like this interesting structure as well, where they've got organelles like the magnetosome magnetic bacteria before, except these organelles have DNA in them.
Yeah, they do these cross sections, cross sections of the bacterium. Yeah, the cell, and then staining for DNA, but then also for membrane. And so then they can see like, oh yeah, these are little inclusions, and they give it a name. They call them pepins.
Yeah, pepins, pepins, pepe. Yeah. Got like, so you've got this thing under the microscope that they've got this really long structure that's like, so they're growing out of mangrove leaves, and then they kind of have this very, very long like cell structure that has that essentially in their genome they've lost their ability to divide their cells. So a lot of the genes that are essential for dividing cells have disappeared, and instead they've just got the cells that make them longer and longer and longer.
Yeah, so yeah, the researchers have access to the genomes of these bacterium, right? They're able to grab a bunch and then do sequencing and assemble something. Oh my God, this I love this visualization.
This is like a visual. Sorry. Oh gosh. I'll play that again because I realized I left Skype on. But yeah. So essentially, you've got this like really cool thing where they start budding off right at the end. So it feels very much like a fungi sort of thing to do.
Hmm.
And
Yeah, make a big cell, and then daughter cells get butted off at the end of it. I think they also drew parallels to some streptomycetes, maybe something like that, like, and some other filamentous style, yeah, filamentous style organisms.
yeah, and here is a look at the structure, and these blue dots are where the genomes are. I think these are the pepins, the
Hmm.
organelles that contain the various like genomes for this
Yeah,
giant microbe.
yeah,
And yeah, just to put in perspective how large, because it needs all this DNA in it to keep it running. Because I think like even like most eukaryotic cells aren't 1 centimeter big.
yeah.
So
I mean, this is kind of crazy. This is kind of crazy from the perspective, like, why does it need DNA in all these different places? How does it regulate stuff if the genome's all like, right, separated? Is it a whole genome in each section? There's many questions that this
yeah,
brings up.
like
Maybe it's not even that. Maybe the genome's only in one place, and these are like structural. Because remember, DNA is also can be a structural thing, strangely enough.
yeah.
So there could be that. Yeah.
Yeah, so this species called Thiomargarita magnificens or something. Not magnificence. Let me double check this quickly. Magnifica.
Yeah. I
Sorry,
just.
Thiomargarita magnifica. So
Uh-huh.
thio means sulfur. Margarita, I think, means pearl, because I think the original like species they form like sulfur pearls, and then like magnifica because it is really really big.
Mhm.
Yeah,
Yeah. I guess another thing that I thought was interesting is like, yeah, these are sulfur style bacteria, so they also have granules of sulfur inside of their cell bodies.
yeah,
Um, and yeah, I mean, we were saying before, like, it matters in science. It matters what you're looking at, right? Because it's difficult to build the methodology in order to make accurate observations of the world. And this is one of those. There was discoveries of giant. We were saying earlier, there's discoveries of giant viruses, and that discovery then led to finding that there's lots of different types of giant viruses. And so, it's studies like this that sort of expand people's horizons and what to look for that potentially then drives new discoveries. I mean, this is a new discovery, but like it could just be the first of many, is what it might be.
yeah. I think that this is because you don't usually look for bacteria this big, and usually
Mhm.
when you see something like this, you'd assume it's a fungus. But they did a couple of
Yeah.
different images where they used fluorescence in situ hybridization to test directly for bacterial DNA.
Yep.
They know it's like related to other viruses, and I think they've. I mean, it'd be really interesting
Bacteria.
if they managed to get to start growing this in the lab because I think there's a lot of really interesting like questions about how life evolved that could be answered here because what we've got here is like
Sure.
a proto-nucleus developing and
Yeah.
there's a lot of
Or I
questions
don't know.
over
I was going to say, not even life evolved, just like how, like, as I was saying, these questions of like, how does a cell like this survive if it has its genome in all these different places, right? Like, it seems wild to me.
yeah I mean yeah the questions of like what because all bacteria have to solve problems and the question is like what is the structure solving for
Ah,
bacteria or what is the
yes, yes.
and that's some and like I mean obviously like I'm thinking also in like kind of terms of like how eukaryotes like so because these have become giant and now they're enclosing their DNA in a material. It's almost like a
Uh-huh,
protonucleus. So question is
uh-huh.
how has that solved it? What kind of how is it different from eukaryote? Because I think there is this theory that
Right.
eukaryote organelles are descended from giant viruses, and it'd be really interesting if
Uh-huh.
these had a similar sort of lineage, or if they're completely different. That'd be both be
Yeah.
really interesting for that perspective.
Absolutely, but but that's all just our thoughts, our hot takes on this particular thing, and we'll just have to see like what appears in the literature in the
Yeah,
future.
and an exciting future it will be.
Okay, so with Yeah, with that, we want to tell everyone to join us next week. If you're into this discussion style that we are going, we'll have more microbiology news for you next
Yeah,
week.
we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter at microtwjc. We both believe that peer review is a process and that we can all participate in it. So again, if you've had a good time listening to us ramble on about microbiology today, or if you think you have something to add, just let us know.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. Bye.